So good morning, everyone. Thank you, Ken, for the invitation. I'm super excited to be here on this beautiful campus and meeting you all. So yesterday we heard some applications of functional MRI. They were mostly in the cervical cord. Dimitri mentioned a few applications in the lumbar cord. But today I will talk specifically about, I will draw some comparisons between the cervical and lumbar regions, highlighting what is easier, what is more difficult in the lumbosacral cord, and what you should consider when setting up an fMRI study in this part of the spinal cord. So on the day before yesterday, we briefly covered the anatomy. So here, or we covered it in more detail. So here, just very briefly, the lumbosacral cord, which is really the lumbar and the sacral part of the spinal cord together, is the lowest part of the spinal cord. Here you can see a schematic on the left and a corresponding T2-weighted sagittal image of the same region. So you need to memorize two anatomical landmarks that we often use. One of them is the so-called lumbosacral enlargement. You can clearly see it here. So this is this is similar to the cervical enlargement, but this is the region which innervates the lower limbs, right? And you can also see the lumbar enlargement on a sagittal T2-weighted image. So this is one of the landmarks we are going to use. And what you also notice that below the lumbosacral enlargement, the spinal cord goes into this cone-shaped structure called the conus medullaris, where the spinal cord decreases in size quite rapidly. And after a short distance, around 5 centimeters, the spinal cord ends, which just going to call it the tip of the spinal cord. And below the tip of the spinal cord, we of course have the bunch of nerve, or a bundle of nerves, which we call the cauda equina. So this is a cross-sectional view of the spinal cord on a T2-weighted multi-echo gradient echo image. The important structures that you see is the spinal cord outlined by the outer red, green circle, and in the middle the gray matter. What you also notice is that the delineation of the spinal cord is sometimes difficult in the lumbosacral region because the nerve rootlets go parallel to it and they have similar contrast. So sometimes it's not easy to tell the boundary between the spinal cord and nerve rootlets. Yes. And the gray matter is often divided into 3 parts: ventral, intermediate, and dorsal horn. So the first difference between the cervical and the lumbosacral fMRI is that we have to use different coils, right? For the cervical cord, we can use the head and neck coil. For the lumbosacral cord, we use the spine coil. The result is that we have to deal with a lower temporal signal-to-noise ratio. Of course, the TSNR is very important to detecting the effect sizes. We want a high signal-to-noise ratio. And here I brought you an example of a typical TSNR map from the same subject. Left, the lumbosacral cord, right, the cervical cord. Of course, the TSNR
Recording in progress.
It depends on the acquisition parameters and post-processing steps. This is the TSNR computed on the raw images. And the resolution was 1 by 1 by 5 millimeters. So you also noticed, first of all, that here I use the same intensity scaling, that the TSNR in the lumbosacral cord is around 50% lower compared to the cervical cord. In both regions, the TSNR is the highest in the spinal cord, lower in the CSF. And you will also notice that the gray matter is a little bit higher than in the gray matter. Another interesting thing is that in the lumbosacral cord, the TSNR increases as you go away from the lung, so toward the bottom of the spinal cord. And in the cervical cord, it's the opposite. So it increases if you go away from the lung toward the head. Here, I just want to show you that we are very lucky that the lumbosacral cord is where it is. If it were a few centimeters higher, then we would have big problems with the image quality. So here you see multi-echo gradient echo scan of the whole spine. And what we often see is strong ghosting-related, breathing-related ghosting artifacts. at vertebral levels T9 to T11. This corresponds to the lower edge of the lung. And below this region of the lumbosacral enlargement, you typically have good quality, but just a few centimeters above, we often see this. So we are quite lucky that in the lumbosacral enlargement, we get okay quality most of the time. Of course, the lumbosacral cord is also very small compared to the brain. Here, more interesting is a comparison between the cervical cord and the lumbosacral cord. And what you have to know is that the lumbosacral cord is smaller in terms of the spinal cord area. So here, this is the same subject. You can see that the lumbosacral enlargement is around 60 square millimeters large compared to the cervical cord C2, C3 level, which is around 90 square millimeters. On the other hand, the gray matter is actually much bigger in the lumbosacral enlargement. So as you can see here, in this subject, it's around twice as big, right? So one can argue that with fMRI, we care about the gray matter, we care about the neurons. So this would actually make lumbosacral fMRI easier compared to the cervical cord. Of course, if you go more toward the inferior slices, the conus medullaris, the spinal cord again gets very small, but the gray matter is still relatively big, right? So the gray matter to spinal cord ratio is actually the highest in the conus medullaris. Another advantage of the lumbosacral cord is that the spinal cord is relatively straight. So we saw during the live demonstration this typical cervical lordosis that we often have to deal with in the cervical cord. This is not the case in the lumbar cord. So with proper positioning, I'm going to show you, this is typically not an issue, and we don't have to play a lot with the angulation. Motion in the lumbosacral enlargement, I think it's quite comparable to the cervical cord. Here you can see an example from both regions. You notice a movement along the phase encoding direction, AP anterior posterior direction in both cases. In my experience, it's quite comparable. Of course, we have a lower signal to noise ratio in the lumbosacral cord, so it might impact performance of the motion correction algorithms. So the first applications of fMRI in this region appeared around 20 years ago. Mostly, I found 4 publications, 3 of them from Patrick Stroman and colleagues, and the other one is Michael Moffitt and colleagues. Of course, these studies were quite preliminary. They didn't have advanced spinal cord processing tools, so they couldn't do, for example, group level analysis. And these studies also use Spinal sequence, so-called seed contrast. Only around 20 years later, the first gradient echo EPI studies appeared in the lumbosacral cord. For now, I'm going to focus on the task fMRI studies. 2 studies from the group of Dimitri and Grégoire Courtine. The first one was done in 3 patients. The second one was a larger, healthy cohort. These 2 studies focus, or the motivation for using fMRI was to help preoperative planning for epidural electrical stimulation. So they wanted to map the segments of the spinal cords that innervate certain muscles, which would help the placement of the electrodes. Then 2 studies from my group in Zurich. The first of them focused on general feasibility and the impact of echo time with a motor task. And the second one focuses on electrical stimulation of the tibial nerve. And I also found another publication, a recent one from Pittsburgh, which again used fMRI to evaluate the impact of neurostimulation. So you notice a pattern here. Most studies used fMRI in the context of neurostimulation, which is different from cervical cord fMRI, where the main motivation seems to be the pain, investigating pain mechanisms. At Balgrist, in my group, we work with spinal cord injury patients, and the lumbosacral region is very relevant in these patients because this is typically below the injury, right? So what happens after an injury is that this part of the spinal cord gets either partially or completely disconnected from the brain, but the neurons are still most of the time morphologically intact, right? But their function is impaired because of the lack of input from the brain. fMRI could be used on one hand to investigate the preserved function below the level of the injury, so we could probe the function of those neurons below the level of the injury, and this region is also often targeted by neuromodulation and neurostimulation, which can. The idea is that it can partially restore then the normal functioning of these neurons. So here you can see a setup for lumbosacral fMRI that we use. Two important things: you have to put a foam wedge below the leg, so you can minimize the lumbar lordosis. So the idea is that we flatten the back as much as possible. The second thing is that we want to immobilize the legs and the lower spine region as much as possible. For that, we use a vacuum cushion and different straps. The lumbosacral enlargement or the lumbosacral cord is around 5 to 10 centimeters long in the rostrocaudal direction. So it can be covered by, basically we don't need a lot of slices to cover it, depending on the slice thickness. We use 5 millimeter slices, so we need 15 to 20 slices to cover this region. Here you can see example axial slices of a gradient echo EPI scan. And I also recommend to acquire structural images which would have the segmentation, landmark identification, and also normalization. For that purpose, we use multi-echo gradient echo scan, but you can also use, for example, high-resolution T2 scan. The differences between the lumbosacral and cervical fMRI in terms of acquisition is mostly that currently we can't acquire the lumbosacral cord together with the brain, right? So what we often do in the cervical cord, that we acquire it together with the brain, either as a big field of view or two separate field of views, is not possible with the lumbar cord, which has a couple of consequences. First of all, less slices are needed, so the TR is shorter, and there is no need for multiband acceleration. It would be anyways difficult within the spine point. And the other consequence is that a small field of view is sufficient, because we don't need to accommodate the brain in the field of view. So typically there is also no need for parallel imaging. The standard sequence for the lumbosacral cord is a single shot EPI with this field of view. The recommended way to achieve it is through inner volume excitation, and seen as it's called zoom it. I believe other vendors have different names. Here you can see an example of the reduced field of view, the yellow box. And if this is not available, then you can also use two saturation bands. But this is less of an optimal option. It increases the TR, it increases the SAR. Yes. So typical parameters for lumbosacral fMRI is a slice thickness of 3 to 5 millimeters. We use 5 millimeter, and Dimitri uses 3 millimeters. In plane resolution 1 by 1 to 1.1 by 1.1. The repetition time as short as possible. Echo time, I'm going to talk about it. And the acceleration, we don't use any acceleration. Some people use a little bit of partial Fourier. Yes, so the image processing map, I covered it in detail yesterday. So I'm only going to focus on the aspects which are more difficult in the lumbosacral cord, which are the segmentation and the registration to the template. But first, one example of motion correction. Here you can see on the left the situation before motion correction. Here on the right, after motion correction. You can see that we use a two-step motion correction. First, flirt, then the SCT fMRI motion. So you can see that the motion along the anterior posterior direction is greatly reduced, but there is still some motion, so it's not 100% perfect. To me, this is not my main concern with the technique, but I think there is still some room for improvement here. So the segmentation of the EPI images here, there are a few examples of the mean EPI images acquired at different echo times, and you can see, and also the corresponding structural image, and you can see that the contrast is not so great on the EPI images between the spinal cord and the CSF. We talked about yesterday. We talked about the EPI-seg method. That method also included some lumbar images in the training set, but we tested it on an independent data set, and we found an okay performance around the lumbosacral enlargement, dice coefficient around 0.8, which means we typically have to still correct it a bit. But it really breaks down if you go to the lower levels, right? Because even manually it becomes really difficult to segment it. So you can see that toward the inferior slices, the dice coefficient goes down. The other tricky thing is the normalization to the template. We mentioned several times that the vertebral levels are not good anatomical landmarks in this region. You can see here, for example, that the neurological and vertebral levels do not correspond to each other. But that alone wouldn't be a problem. The problem is that this correspondence itself, or the relation of these two levels, is also variable across participants, which you can nicely see here. For example, the LSC, so the lumbosacral enlargement, can be anywhere between T12 and L1. Also, the length of the lumbosacral cord can vary between participants between 30 to 60 millimeters. Right? So the current standard or state of the art that most group, or what we use for normalizing these images is the spinal cord toolbox. So the spinal cord toolbox implemented recently the spinal levels, which are based on the frost cell atlas. So there is this study. It's a review that collected based on different studies, came up with this correspondence between the neuronal segments on the top and the vertebral levels. But these are just approximate neurological levels, right? But the idea is that once you can normalize your images to the PAN50 template, then you can see these approximate neurological levels for orientation, right? For normalization to the template, we use two anatomical landmarks: the slice with the largest cross-sectional area, or the center of the lumbosacral enlargement, and the tip of the spinal cord. On the EPI images, it's very difficult to identify those landmarks, but on the structural images, it's much easier. So here you can see on top, this is the slice at the middle of the lumbosacral enlargement, the slice with the largest spinal cord area, and here you can see the tip of the spinal cord. You can identify it manually when you go through the slices. And then during normalization, you can register these coordinates to the corresponding coordinates of the PAN50 template. This works quite well in my experience. The disadvantage is that it doesn't take into account if participants have different distributions of segmental levels, right? Or of spinal levels. So it can be, for example, that somebody has a lower, I don't know, S1 neurological levels than others. So ideally, we would like to register we would like to identify in the native space the individual spinal levels. This has been done in the cervical cord. So there is this nerve root-based segmentation available. This was trained only on the lumbar cord. In the lumbosacral cord, this task is more difficult. You can still see the nerve rootlets. Here you can see an example of the rootlets in the spinal cord. But visual identification is quite is quite challenging, especially not so much in the cauda equina and the sacral levels, but more at the higher slices where these nerve rootlets join together. You see, for example here, and it becomes very difficult to see where they actually enter the spinal cord. But still, two groups attempted to do that. So the first study from Dimitri's group even provided a recipe how to do that, but just very quickly, first they find the T12 rootlet. So this is the rootlet that exits just below the T12 vertebra. They track it, the rootlet, all the way back to the spinal cord, which is then the T12 segment spinal level. And then they know that, okay, then the next rootlets that exits the spinal cord must be L1 and so on. But the point is that it's not very easy. Depending on the data quality, it would not even be possible for every rootlet and for every subject. Moving on to the applications of lumbar fMRI, when we started working with this technique, then the first application we did, we just went for the most robust task possible. So we asked participants to move their ankle up and down, active movements. We asked them to do it in a repetitive way at a frequency of 0.5 hertz, and we used a block design. So 15 seconds movement, 15 seconds not movement. We did 2 runs of 10 minutes each. And this is the group level activation that we observed. So the first thing that you notice is that the activation in response to the active movement is extremely lateralized on the ipsilateral, in this case, the right side of the spinal cord, and also the rostrocaudal distribution corresponded to what we expected. In the cross section, it was also what we expected. So you can see that activity, which very well corresponds to the ipsilateral ventral and intermediate horns. So we were very happy about these results. It seems that on the group level, at least, fMRI can provide very spatially specific results. Then we went on and looked at the subject level results, where the picture was less clear. So here you can see the same slides across different subjects. Everybody did the same task, but you can see that there is a large variability both in terms of the amplitude and the location of the activation. There is still some trend that we observe. So the activation seems to be in most cases on the right side and in the ventral portion. But as I said, there is a lot of variability on the subject level results. Somewhat reassuringly, we repeated the experiment in subjects who did not perform the task. Everything else was the same. And in those 3 subjects, we did not observe activation, or the false positive rate was 1%, which was a nominal false positive rate. So it seems that the activity we observed in the subjects with the task are really related to the task, because every single one of them we observed the activation, or the rate of significant voxels was way higher than the false positive rate, but there was this large variability. So the large variability across subjects was recognized already 20 years ago in many studies. Here I just mentioned a few, and several hypotheses were proposed about the reasons. One of them is the anatomical variations in the gross vascular architecture. So here you can see, we know that the gradient echo EPI based fMRI has a large contribution from the large veins. So it seems plausible that the signal that you see depends on the vasculature. So here you can see a posterior vein just behind the lumbar cord, and you notice how complicated the anatomy is. So it does these zigzag movements, which are highly variable across subjects. Here, these are the axial images. You can see here in the middle a posterior vein, and you can see how it moves to the left, to the right. Sometimes it goes more away from the spinal cord. Sometimes it goes closer to the spinal cord. Here you can see it once again. That's the vein. So I think it's plausible to think that this anatomical variability in the veins also explain part of the variability on the subject level results. Other reasons include differences in the baseline physiological state, especially the blood volume, blood flow, vascular reactivity, differences in level of arousal and task performance. In Julian's study, he investigated the relationship between end-tidal CO2 level and the bolt signal change, and he found a negative correlation. So basically, the lower the end-tidal CO2 was, the higher was the bolt signal change. We also looked at whether the data quality is somehow related to the bolt signal change. So here we fitted a linear fit across 100 runs, and for each run we computed the percent of outlier volumes as a metric of general data quality, but we did not find any significant relationship between the data quality and the bolt effect size. We also looked at the optimal echo time in the lumbosacral region. So we repeated the task, the motor task, at 4 different echo times: 20, 28, 35, and 42 milliseconds. We did that within the same sessions. The order of the echo times was randomized across subjects. And here the picture was very, very interesting. So here you can see in the different columns, the different echo times. And the first observation was that the echo time of 20 milliseconds was clearly the worst. The higher echo times gave more activated voxels, much more activated voxels. The bolt signal change was actually the highest with the echo time of 42 milliseconds. We also looked at the split half reliability, so we divided the data into 2 10-minute parts. Then we looked at their similarity using the dice coefficient. We also found that the dice coefficient was higher when we used the longest investigated echo time, 42 milliseconds, which is interesting because, of course, the TSNR was the lowest for the highest echo time. We looked into literature, and there are a few studies which could help explain these findings. So Rob Berry found, for example, that the T2 star in the cervical gray matter is around 41 milliseconds, which nicely corresponds to our results. One brain study also found a peak of the bolt response at 40 milliseconds, but of course the peak is very broad, right? So I think above 30 milliseconds, if you're between 30 or 40, it doesn't make a huge difference. But the 20 milliseconds clearly seems to be not optimal. Another interesting observation is that this large unspecific activation we observed at 20 milliseconds, but also at 28 milliseconds. So this is in the anterior medial part of the spinal cord. You can see it here. And at 20 milliseconds, this was even stronger than the specific activation in the ipsilateral ventral part of the gray matter, where we actually expect a neuronal activation. But at the highest echo times, this activation actually disappeared. We also often see this activation in studies that use motor tasks. So here, one example from a study that used knee extension. Again, you can see this spatially unspecific activation in the middle of the spinal cord here in both slices. We think that it's related to large vein effect. So we know that a large vein that drains the ventral gray matter horns is located just right there. And we also know that from a brain study that the part of the bolt signal which comes from the vasculature peaks at a lower echo time. Here in the brain, it was found to be 21 milliseconds and then becomes less afterwards. So in our opinion, this is a large vein effect, which is not a false positive per se, but this is also not something that we are interested in typically. The next study I would like to talk about is from Dimitri's group. So they applied not only active movement, but also passive movements of lower limb muscles and also tendon vibration. The tendon vibration is useful because you can access different muscles, also muscles which are difficult to activate within the scanner, for example, knee, hip extension. And yeah, so here's basically the setup: active movement, passive movement, and tendon vibration. They tested different tasks. And on group level results, at group level, here you can see the results. They observed, on one hand, a rostral-caudal distribution between the different muscles. So, for example, here you can see that the activation or the knee-related activations appeared higher than the ankle when stimulating the ankle. And they basically said that the active movement tasks seem to be the more robust in terms of correspondence with the anatomical expectations. The second robust was the passive movement, and the least robust was the vibration task. They were also very interested in subject level results because of the motivation of the studies. Basically, they wanted to obtain personalized connectomes, so in each subjects map the segments of the spinal cords that innervate specific muscles. Of course, it's very difficult on the single subject level. They came up with a recipe how to interpret the subject level results better. I'm not going to go through it in detail, but basically they look at aspects, for example, okay, what is the size of the cluster that they see on subject level. Where is it located? Is it more located on the dorsal, on the ventral side, on the ipsilateral, contralateral side? And based on these considerations, you can have a confidence whether the seen cluster corresponds to certain muscles, right? And using this scheme, they mapped the clusters corresponding to different muscles on a personalized 3D models. What you can see is that it's quite variable across participants, and they concluded that delineating reliable innervation map for all muscles and participants is still challenging, but more robust acquisitions would help. But they provided a recipe for how to make sense of these subject level results. In my lab, we also tested electrical stimulation. Electrical stimulation is useful because we can access different nerves in a highly controlled way. It's also very relevant for spinal cord injury patients, where electrophysiological measurements are often used. So here we also went for a block design: 15 seconds stimulation, 15 seconds no stimulation, and we stimulated a peripheral nerve on the one side, on the right side, the tibial nerve. And here these are the group level results on 25 patients. You can see again a highly lateralized activity on the ipsilateral side, but this time the activation was more on the dorsal and intermediate side of the spinal cord, of the gray matter. Here you can see it clearly, which is in line with what we expect, because we know that electrical stimulation is quite an unspecific activation at the high amplitude that we applied. So basically, it stimulates everything, the A beta fibers, C fibers, A delta fibers. Then we also looked at whether we really need the 40-minute scan, 4 times 10 minutes, to obtain this group level activity, or we can also achieve that using shorter or less data acquired. So here, basically, what we did is that we started reducing the data, and we did the group level results, and we did different combinations. So basically, for example, in these data points, you can see the number of significant voxels if you only used 2 runs, 8 minutes each. And here, if you used all 4 runs, but 8 minutes each. So one thing that you notice is that the number of significant voxels goes up if we include more data per session, but it largely plateaus after around 24 minutes. So it doesn't seem to be needed to acquire more than 25 minutes of data, which I think is good news. The other thing that we notice is that there is not much difference based on how we allocate the data. So for example, if you look at these 2 time points here, this is 3 runs 8 minutes each. This was 4 runs 6 minutes each. In total, the same amount of data and pretty much the same number of significant voxels. And we found one study that investigated the effect of amount of data used per session, but they've had different findings. So it was Can study from 2016. They used thermal stimulation, and they found that the more data you use, then the number of significant voxels goes up linearly. So there was no plateauing involved. And even more strikingly, when we did a split half analysis, we looked at the first 10 minutes of the acquired data and the second 20 minutes, sorry. The second half of the measurement returned no significant voxels. Yeah, okay, so, which was again different from Can study. We found that actually later runs linearly increase the number of significant voxels, which strongly implies that with the electrical stimulation, we are dealing with habituation effects, and with the thermal stimulation, we are dealing with sensitization. So I think it's very important when you set up a new study, consider these things, right? And we even went further and repeated the experiment exactly 7 days later. So the scan was done on the same day of the week, the same time point of the day. And here we also found an interesting thing. So basically the second time, when we repeat the scan, then the overall activation on the group level was much weaker. So here we can see the results on session 1, the results on session 2. And individually, both activation maps make sense. They are kind of in the right location, what we expect. But during the second session, it was just much weaker. So this is something that we still don't understand. We dug a little bit deeper. What could be the reason? So here you can see the ball signal change across the different runs of session 1 and session 2. And what we noticed that during session 2, the first run had a quite comparable effect size compared to the first run, session 1. But right after the second run, the effect size rapidly declined. So it could imply, we are still not very confident, but it could imply that the habituation kicks in much faster during the second session. Now we are actually acquiring the data a third time, half a year after these scans, just to understand a little bit more the test-retest reliability, long-term test-retest reliability. So I think I'm just going to go through very quickly the resting state results in the spinal cord. There are a few studies. I'm aware of 3 studies that investigated fMRI in the lumbosacral cord, and they investigated different aspects, which parts of the gray matter are correlating with each other, what's the effect of scanning parameters, what's the effect of denoising parameters, the denoising techniques, what's the test-retest reliability. But what no human study investigated yet is the physiological origin of these correlations. And here you can see that the challenge, when I think about the resting state fMRI, I always think about gray matter segmentation, because it's so difficult to do, especially in the lumbar cord. So the 3 different studies all use different techniques. First, use the atlas-based, so warping the PAM50 atlas to the native space with manual correction. The second one directly segmented the gray matter in the native space. We segmented the anatomical image, put it on the EPI and corrected it. But basically every study used manual segmentation at the end. Just real quickly, the strategy that people used is that they identify the LSC slice and 2 slices above and below. The intersegmental connectivity is what's already being done in the cervical cord, has not been done in the lumbar cord yet. Just real shortly, so the effect of denoising on the resting state results is very profound. So here you can see the distribution of gray matter voxel to voxel correlations before denoising. That's the red distribution, and after denoising it becomes much narrower, which can be seen also here. So before denoising, everything correlates with everything in the spinal cord, but after denoising, the correlations become much less, and some interesting patterns appear. I think for the sake of time, I'm going to skip these slides. Here, just very quickly, we also include a region of no interest. This is a pair of regions which are not supposed to correlate with each other. And for that, we use the ventral and the dorsal white matter regions, which are supported by different arteries, drained by different veins. So we don't expect a coupling between them. And the typical pattern that different studies found in the lumbosacral cord is similar to the cervical cord. The highest correlations between the bilateral ventral horns and the bilateral dorsal horns. And the other correlations were much, much lower. This is also what you can see here on these Roy to voxel based correlation maps. Finally, we also looked at the test-retest reliability of the resting state maps. It was the same study, so the time frame was 7 days. And we also looked at the reliability when we use the different lengths of acquisitions. And the first thing that you see is that the 6-minute scans did not show the highest reliability. 8 and 10-minute scans showed the higher test-retest reliability. And the reliability was the highest for the bilateral ventral correlations, which 0.6, almost 0.66, which I would consider it a very good test-retest variability. For the other correlations, the reliability was considerably lower. Similar observation from Merve's study from the cervical cord, where the ICC was, I think here the difference was 1 day between the two experiments. The ICC for the ventral correlation was above 0.6, quite comparable. But they also found a higher ICC for the dorsal correlations, which for us it was not the case. So to sum up, the lumbosacral cord has both advantages and disadvantages compared to the cervical cord. What is more difficult is the lower TSNR, but on the other hand, we have a larger gray matter area and the straight spinal cord. What is more difficult on the processing side is the segmentation and the normalization. And the current literature seems to be motivated by the need to understand the preserved function below the spinal cord injury and to support neuromodulation strategies. So final slides, we look at spinal cord fMRI currently as a research tool that can be used on group level studies to investigate motor and sensory pathways. But of course, ideally, we would like to use it as a clinical tool to assess residual function after injury, investigate effectiveness of therapeutic interventions. This need was already recognized almost 20 years ago. From this study, the goal is for spinal fMRIs to be useful and practical clinical tool. At the moment, we are not there, so we need quite a lot of improvement in terms of robustly detecting both responses on a single subject level and to achieve a better test-retest variability, which could be done by improving image acquisition and image processing, especially denoising methods. So sorry if it was too late.
All right, awesome job. Very thorough, really good. Enjoyed hearing about the challenges and your solutions to them. I'll start off with one question, and that's looking at your habituation effects that you see. And curious whether those could also just be training effects. Is it habituation, or is it that they don't have to think as much to do their dorsiflexion, plantar flexion, maybe?
Yes. So we didn't look at the habituation for the motor type.
Oh, okay.
Only with the sensory task, but we noticed that the participants didn't report any change in the sensation, right? So they seem to experience the same sensation, but still the effect was very clear, right? So the second part of the measurement contributed nothing to the group level results. And I mean, we expect some sort of plateauing alone from the, I think, statistical point of view. I think that if you acquire more data, that at one point you don't really get much, I think. But in this case, it was really striking. So I think it's more than the statistical effect.
Yeah.
It's really happy to mention.
Okay. Thank you for clarifying that. And then in my study where we looked at, patients were experiencing more pain over that course, but I didn't collect the data, so I wasn't able to quantify that. That's an anecdote. All right, questions here?
Well, it's more like a comment. So for the habituation, I think can you also see perhaps maybe some habituation from electrical stimulation? And I know that Gary has done some brain fMRI with the same stimulus.
Yes.
Also see some habituation.
Yeah.
So I think maybe we need to do a combined brain and spinal cord.
Yes.
And see what happens.
But the same thing was observed in the brain too, brain fMRI.
Yeah, we just definitely see habituation, and it's a function of the pulse rate, the PRF, all of those things.
Yes.
Can markedly change the habituation as a function of time.
Yes.
So it's pretty complicated. And as you showed, small changes in the process can make larger changes in what happens as a function of time and day and so on.
All right. Any other questions? Yeah.
Thank you, Gergely. Very instructive. About the processing and more specifically the segmentation of the gradient echo EPI.
Yes.
I was wondering, have you considered not segmenting it and just relying on the registration to the anatomical multi-echo GRE?
So basically we would register it to the PAM50 template.
To the multi-echo GRE of the PAM50 plant, right? And then the multi-echo GRE to the PAM50.
Yes. So basically that's how we segmented the spinal cord. So we segment the spinal cord on the anatomical image and register it to the EPI.
Okay. So you did not bother to get the segmentation on the EPI?
Yes. No. That's why we acquired this.
Yeah, I think it's good.
Yeah. But the segmentation on the structural image is still manually.
Okay. So
yes.
All right. Questions? Questions online? Okay, I've got. Well, I'll go next.
Yeah.
Your interest in doing the nerve root segmentation in the cauda equina. So we're really interested in that for being able to better assess stenosis in the lumbar spine. We're going to be collecting a full spine data set very soon that we'd be happy to share with you and collaborate on a project. So we're happy to make data available.
Yes, so I think we should also consider what is the best sequence to acquire or to visualize the nerve rootlets. So one study comes to my mind from the Chinese group who even put their data online, and they compared 3 different scans: the T2 weighted TSC, a DES sequence, and a CIS. These are Siemens terms, and they found that the CIS sequence has the best visibility. This is the image that I showed you for the vasculature to show that.
We probably still want to do it on T2 weighted because that would be most clinically relevant, I guess, for our needs.
Yes.
But it would, talk more. Oh yeah. Go ahead.
I mean, I was curious about the the question of how many rounds or how much data you need. So it seems to be 25 minutes, and so I was just to come back to what Alexandra Timmerman was describing, that she found that having a longer run is actually better.
Yes.
And so if I remember correctly, you had like 2 times 10 minutes, and this was less good Yeah, so I was just wondering if you try to address that hypothesis, number of runs versus length of runs. So the 2 times 10 is not as good as, I mean, obviously there's a bit of noise, right, in the process, but
Yes.
not as good as I get.
Not as good as which one?
Yeah, well, you have like an equivalent of 20 minutes, but let's say I don't know, if you try like like
yeah.
multiple runs, like if you have 4 runs, that you make shorter, like 5 minutes, let's say,
Aha,
and like
aha.
5 times 4, just to see how
Yes.
it compares.
That we did not check. So the minimum duration we use is 6 minutes, but here we could check
Just to,
4 times 5.
yeah,
Right?
just to see how it compares to what Alexandra was saying.
Yes, yes. But I mean, for us, this is the best comparison because it's exactly 24 minutes, right? And the difference is so small, so it doesn't. But again, it's like 6 minutes versus 8 minutes, so it's quite different, similar. In terms of.
Right, and but were they a painful stimulation?
It was not very painful, but it was also not very comfortable. So they definitely felt it.
Okay.
And we know that kind of stimulation already activates the A delta and C fibers, so the nociceptive pathways. But on a pain scale, 1 to 10, it was maybe 2. Yeah.
Okay.
Was,
okay.
yeah. I mean, it was something that
how
they,
it compares
they had to. Yeah.
what you saw. Yeah.
It was something they had to endure for 40 minutes, so it was not.
Okay. All right. Thank you.
Okay. Any other questions? I have some more. Curious about patient size, and do you have any limits on the size of patients? Just because I assume, depending on how much subcutaneous fat they may have, that they're going to be much farther away from the spine coils. And have you?
Yes. So we definitely noticed that the further away we are from the spine coil, the worse the quality gets in terms of TSNR. Yeah, I mean that's something we can do about, but there's definitely this effect, right?
Yeah. And we looked a little bit about also including an anterior array, like abdominal array, and it doesn't seem to help.
I tested that too, and it made no difference. Yeah, so we don't use it.
Okay. All right. Thank you.